Hi everyone, thank you for joining us today. My name is Mylene Simon. I'm a software and system engineer, and I'm the lead developer of the project that I'm going to present today. I work at NIST, so this is the National Institute of Standards and Technology in the US, but I work as an associate/contractor, so I am not a federal employee, and what I will say does not represent the US government, so just a standard disclaimer. Yeah, and I think it's 10, so I will slowly get started to give a chance for people to jump in. Try to go to the next one. Yeah, and another kind of standard disclaimer that we put at NIST. So I may talk about some commercial equipment, commercial software, for example. This does not mean that NIST officially endorse what I mentioned. So what I say is my opinion, and it's not representing the US government. And in terms of what we are going to talk about today, so I'm going to present our in-house open source platform for image analytics. So it's called WIPP, for Web Image Processing Pipelines. So we'll kind of go through a full slide to introduce the platform, the concept of plugins that we have in the platform, present the scalable visualization that we have, and kind of talk about how we integrate AI as a service in this platform. And then I will move on to more of a demonstration. So I will go to the platform and run a few example workflows, see how we can upload data, how we can configure jobs into chained computational workflows, and then look at also the scalable visualization with some example data sets. And to conclude, I will talk about what we are doing right now. So the work that is currently being done and implemented in the platform. So it will mainly talk about how we are trying to implement easy AI retraining in the platform and how we are interfacing with image annotation platforms to enable that. Just to kind of talk really quickly about NIST, if you don't know the institution. So this is a federal lab in the US. So it stands for National Institute of Standards and Technology. And so it has mission to promote innovation, industrial competitiveness, advancing measurement standards. So a lot of standards are made, are worked on at NIST. And yeah, so there have been a few a few Nobel Prizes coming from NIST. And we have multiple laboratories in NIST. So this project WIPP is part of the Information Technology Laboratory, so we call it ITL. And this lab focuses really on IT measurement, testing standards, and we specifically focus on reproducibility of scientific computation for metrology. And we design computational environments, data and metadata formats, and APIs to reproduce measurements. So to introduce the WIPP project, so why did we, let me just reduce that, yeah. So why did we started to implement this platform for image analytics? So the goal was really to create a web-based platform for scientists, so that would be easy to use for scientists that could access the platform through the web browser, and they will be able to access, process, visualize, and interact with large-scale image collection. And it really started because we were working a lot with biology, so they had a really large collection of microscopy images, and then they will be stitching a lot of field of view into a really large image stitched together. And so it was starting to be complicated for them to visualize the data, process the data. So we really wanted to build a platform that would be easy for them to interact with the data, but also offload the computation to servers and clusters. And so, yeah, so the goal is to achieve high statistical significance of image-based measurements, and also to deliver traceable measurements over large image collections. So we really do care a lot about traceability in this platform and this project. And yeah, we want to support reproducible research over terabyte-size image analysis. And so we try to gather really provenance information about all of the data and computations that happen in the platform. And we try to incorporate many data and metadata standards or best practices, and to build interoperable software. And as I mentioned before, so we work a lot with biologists and other material scientists. So we focus on microscopy image analysis mostly. Some of the challenge when we build the platform, obviously the challenge of big data, so data becomes bigger and bigger, so especially microscopy as an instrument evolves, we get more data more quickly, and then we have to think about infrastructure, so how to keep it low cost. And as I mentioned, traceable and reproducible computation are really important for us, and how we can make sure that when you get a result, then you can go back to all of the steps that were used to produce these results. And this goes to trusted computation, secure, because if you work with medical images, for example, you might want to really make sure that the data is not accessible by anyone. And also, if you are working on a project, a research project, you might want to keep your data for yourself before you publish. And we wanted to really have a system that is modular and interoperable, so we can kind of plug multiple container-based plugins, so computational plugins into the platform, and hopefully also reuse these plugins across other platforms. And we added interactive visualization, and I will demonstrate all of that later. So let's move on to WIPP. So what we kind of call the WIPP ecosystem. So we have the WIPP platform, we have WIPP plugins, and then we have a plugin registry to list all of the available plugins for the platform. So I'm going to talk first about WIPP platform. So as I mentioned, WIPP stands for Web Image Processing Pipelines. It is a web-based and scalable open source platform for distributed image processing computation, trusted measurement, and online data exploration. So we do have private workspaces. So whatever you do will be private, and then you have some option to configure the visibility of your data and computations. You can, from the web platform, configure computational workflows, and then you can trigger the execution from the web platform. And the workflow will be using containerized computational plugins. So this is software algorithms that are packaged into containers, so Docker containers, for example. And then this is what will be running on the backend on a server or on a cluster. We do have data exploration tools also. So we have deep zoom interactive visualization for images. And it is AI-enabled in the sense that we support GPU in the platform. We have a TensorBoard integration, and we interact with image annotation tools. So you can do AI-related computation in the platform. And so to enable all of that, we really leverage containerization and container orchestration technologies such as Docker and Kubernetes. And we have a cloud agnostic design. So it means that we can deploy on premises. So for example, at NIST, we deploy it a lot on server or set of servers, but we can also deploy in the cloud. So we have collaborator deploying it in the Amazon cloud, for example. And so what we can do in the platform in terms of computation, so we can create workflows using the plugins that are registered in the platform. So when you get the platform, it is basically empty, and then you can register either plugins that we provide, or you can create your own and plug it into the platform. So for example, here, if we have a simple two-step workflow, so we do training of an AI, and then we use the trained model to infer on some images. So we will get a two-step workflow here. Then we can monitor in the dashboard the execution. So you can click on each step and see the logs, and then you would get the results. So in terms of visualization, so we have a few different options that I'm going to show later on about visualization, but we have this one where we can overlay different layers. If you have different channels, for example, for the same image, or if you have an image and then you have the segmentation and you want to overlay the two of them to check. And then we can apply color to each layer. We can apply contrast, things like that. We can invert also the image. So we can do all of that and then overlay them. And we can also have a time series, so we can move across the time series and check things and visualize. And it is deep zoom. So even if you have a really large image, behind the scene, we use a pyramidal representation of images. So that means that we have also an image server that will render different resolution of the image as you zoom in, zoom out, and pan. So that enables really smooth deep zoom visualization, even if you have a really large images. And this leverage the triple IF APIs, and we use an image server for that is called the double IP image server. And in terms of what happens in the browser, we use a JavaScript library that is called OpenSea Dragon, and this is what really enables the deep zoom visualization. Yeah, so this is another example of images. So this is about 20K by 20K pixel. And so we display it in the platform and you can zoom in and zoom out. So I talked about the platform, but if you want to do computation in a platform, we have to have computational plugins. And so we call them the WIPP plugin, and they are really interoperable containerized algorithmic plugins. So really an algorithm that is packaged into a Docker container. And with that, we associate a plugin manifest that is in the JSON format. And this is where we specify what are the inputs of the plugin, what are the outputs, some information, some metadata about what is the name, version, description, the author, GitHub repository, for example, citation if there is any associated with this code, and also the container ID, so really the container image name, so what you would see in Docker Hub, for example. So you see here an example where we have a really simple computational plugin that will do just thresholding. So we package it in a Docker image. We use Docker file to describe how the algorithm will be packaged in the Docker image. And then we have this JSON plugin manifest that really describes. So this is a thresholding plugin. So it will get a collection of images as an input, maybe a threshold value, because it's a really basic thresholding plugin. And then as an output, it will get another collection of images that will be the result. And then we have some UI entry, because we do generate the forms for the job that the user will configure to create workflows. So we will create them on the fly from this manifest. And then at NIST, we also try to collaborate with other institutes and kind of build community standards. So we have had these efforts in the form of two workshops that we did in 2019 and 2023 to try to come up with community standards and guidelines for scientific-based containerized computational software and trying to make them as interoperable as possible. So we have a GitHub repository, and this is really something that is ongoing. Maybe we might do another workshop and try to gather community best practices and come up with some guidelines that make everything as reusable as possible. And finally, so since we have this notion of plugins that you can add in to your platform, we decided to build a plugin registry. And this is actually based on another project that is developed in our group that is called CDCS, and it stands for Configurable Data Curation System. So this is a system basically where you can define a schema, and then it will allow you to curate data, to have some kind of a custom database with web interface where you can do all sorts of searches and so on. So it's really a configurable system that you can apply to any kind of domain. So we did a custom version for our WIPP plugins. And really this allows us to share the WIPP plugins, but also because it supports federation, it means also that if we were to deploy this plugin registry across multiple institutions, we could then enable federated searches across multiple institutions. So if at NIST we have some plugins and in another lab they have other plugins that are of interest, we could share plugins and all import them all in the platform. In terms of how we use AI in this platform, so there are kind of different ways. So the most obvious one I would say is because you can have plugins that use a GPU, so this is something that is supported, you can have an AI-based plugin and an AI-based code that is in your plugin, and then it will run as part of your computational workflows. So for example, we have some unit semantic segmentation training and inference plugins that we have in the WIPP team. We also have some classification plugins, so they're all listed in our GitHub. But we also we have started to generate plugin that will interact with AI model repositories. So for example, Hugging Face and BioImage.io, which is one that is more dedicated to bio-related AI models. So this is something that we have started to use internally, and we have someone right now rewriting the documentation and doing all of the tests so we can publish them as well for others to use. Yeah, so some example of what we have right now. So we have UNet. We also have a specific Stardist plugin that you can use to segment nuclei, for example. Okay, and so now that I've talked a bit about the platform, I'm going to move on to more demonstration type of thing and looking at how we can create and run computational workflows in WIPP, but also how we can import images, how we can visualize them, etc. So let me just stop quickly to see if there are any questions in the chat. And if I can share the presentation and additional WIPP links. Yeah, so I can put in the chat a few WIPP links at the end. And I think everything will be recorded and posted on the YouTube channel. And then we have I will put at the end of the presentation, we have a WIPP. So it's WIPP-team@nist.gov, where you can send any questions and we can share the presentation also via email. Okay. So let's start with some really simple workflow. So for example, if you have a set of tiles, so you acquired an image with a microscope and you got multiple tiles that you then want to stitch together to get this big field of view, and then you might want to do segmentation and feature extraction. So this is kind of a typical use case. And this dataset is also published on one of our GitHub repositories. So what would be the workflow steps that you would want to do? So you would want to upload your images to the platform. You want to stitch together the field of view, so you get this big field of view. You want to segment the images, and then you want to extract some features. And at the end, you want to visualize everything, overlay maybe the channel and the segmentation results. So you start by uploading the data on the platform. Then you will build the workflow. So this is an example of the view of the workflow. And I'm going to move to the actual demo just after that. So you can see we start by stitching. So we have an algorithm that is specialized to stitch microscopy images. So we run that first. Then we want to actually assemble the images. We might want to do some segmentation. Then we want to do some labeling of the segmentation, extract some features. And because we have two channels in this case, we have the fluorescent channel and the phase contrast, then we want to duplicate some of these steps for the two channels. As I mentioned, one of our first steps in this workflow would be to use NIST. So this is a microscopy image stitching tool. It's also available as an ImageJ Fiji plugin. So if you are used to Fiji, you can use it there. And it's also available as a WIPP plugin. So yeah, so it's really useful to stitch microscopy images. It outputs something like that. So what we call a stitching vector that really specifies the position of each tile in the big field of view. And then from there, we have an assembly plugin that is able to get the list of field of view, this stitching vector metadata file, and assemble the small tiles into a big field of view. After that, we want to do segmentation. So here we have a plugin that's called EGT. It stands for Empirical Gradient Threshold. So we will get then this type of segmentation. you can do some labeling, and then you can overlay all of that into the system. So let me move, so to the actual platform. Okay, so let me check if I am. Okay, so this is the actual platform. So this is one of our deployments that we have here at NIST. So when you go to the home, kind of home page of the platform, you get what we call a quick view dashboard. So you have the latest image collections that you uploaded to the platform or that were created as part of a workflow, the latest visualization, and the latest workflows. So if we look at, for example, some image collection, so if I click on it, I can see some information. So I can see that there are 600 images in this collection. It is in the iterative format. So we do follow the Homero microscopy environment kind of standard for microscopy images. So everything you upload to the platform will be converted to omissive, and more specifically, it will be converted to tiled pyramidal omissive so that we can enable deep zoom visualization on that. And then from the image collection view, you can also look at your images. You can click on each image and then you get a deep zoom view. You could also like kind of make it full screen, for example. And then we have some kind of more UI related stuff where you can either present the images as a list or as a grid. We can also enable contrast. So this one probably don't need much additional contrast, but we could like add some color mapping that we can enable in the view. And we can also invert. So if we wanted to invert for visualization purposes, we can do that. Okay, and so if I look at some other data here, so I have, for example, this is what we call the big data experiment. So this is a data set from a few years back, but it is interesting because the images are quite large, so they are stitched field of view. So this one, for example, is, so both of them are over 20K by 20K pixel. And this is like 2.5 gigabytes. So, but even with that, because we do have the pyramidal representation and the image server behind the scene, we can still visualize it without any issue in our browser and enable the deep zoom like that. And so as you can see here, I have two channels for this image. And what I might want to do is to overlay them. So I can do that in the section multi-channel visualization. And for example, I configured here for this data set. So I have the first channel as a layer, and I can specify that I just want to do some contrast stretching on this one. I don't want to apply any color map or any inversion. And then I have another layer with a second channel. And so that gives me that. So let me make it full screen. So still the same view that we were seeing. But now what I can do is to click on this one. Then both of them are overlaid. And I can adjust the transparency. So this is something that we use also here to for example, if we have segmentation mask, we overlay the segmentation mask, and we can really assess the quality of the segmentation. Okay, so if I exit this mode, let me just show you quickly how we can upload data to the platform. So there are multiple ways. So we can either upload through the browser, and this is what I'm going to show you now. This is the simplest option. We also have some batch import features. So if you have data that is available from where the system is deployed, so either if it's on the servers and the data is available on the server, or if it's in the cloud, it's available through a persistent volume, for example, then you can do a batch import. So it will ingest the data from the backend. But let's just start with something more simple. So So I will create a new collection. I will specify that I don't want to use a backend import. In this case, I just want to upload through my browser. So I will get a new collection created. And here I can see that I can either click on these buttons or I can also drag and drop. So let me do that. Get a few images, so it's not too long. Right, so this is the images that I'm looking at. I have some issues with my mouse. Okay, here we go. So when you drag and drop, so you will see the files that are queued for transfer to the backend, for transfer to the server. And so while they are being uploaded, then you see them appear here. And this kind of animation means that they are converting to the omissive pyramidal tiled format that I mentioned just before. So, and as they get converted, they then appear here, and then we can click on them and get the deep zoom view. Yeah, so basically this is how one of the way we ingest data into the platform. So the more simple way, I would say. And for each image, you still have the option to download it. You can also download the metadata. So this is the only metadata that is extracted from the image. And you can also delete them. You can delete all of them at once. Or you can delete the whole collection. Okay, so let's look at the workflow that I was mentioning before. So we were looking at from a set of tile, doing the stitching and then doing some segmentation, and finally some feature extraction. So I have this workflow already done so that I don't go through all of the steps to configure them, but let me show you. So for each step, we can see which plugin was used, and then we can see all of the inputs. And what it does is that we can click on it and go back to the actual collection. So this is one of the things that we do to enable traceability of the computation. So everything is hyperlinked. So you can, from a job, you can always go back to the inputs. You can go back to the original workflow. You can navigate through all of the data like that. So here we can see input and outputs. So let me copy this workflow just so that I can show you how to add a new task. So yeah, so we have the workflow here. So let's see what we could do on top of that. So we have the phase contrast here. We did EGT segmentation for the fluorescent channel. So let's see if we want to do thresholding, for example, on this one. We want to use a really basic thresholding technique. So we click on Add Task. Then we can select which plugin we want to use. So in my case, I want to use thresholding plugin. So this one is basically just thresholding methods from ImageJ. I'm going to call it phase thresh maybe. Here I can start typing and I will get all of the outputs from previous steps. So this is how I can link jobs together. So in my case, I want, which way do I want? I want the phase, yeah, I want the phase contrast. And let's see, I will do O2, for example. So I can add the task. As you can see, it gets inserted into the workflow. And then what I can do is submit the workflow and I can monitor the execution. So this is called Argo workflow. So it's a Kubernetes native workflow orchestrator. So it will run on a Kubernetes cluster. And for each step, I can basically click on each step and then look at the logs. So we'll get logs from here. So basically here it's mostly reading data. I think it's already done. Yeah. By the time I clicked, it was already done, but let's see another one. Yeah, so still not fast enough to go through it, but yeah, so we can monitor the execution, and if something was to go wrong, then we could see in the logs what happened and correct for the next time. Okay, so while this is running, let me go through maybe what would be the results of that. So let me go to the one that was already done. So as I mentioned, once we have the jobs and the execution is done, we can then go to the output of each job. So here, for example, I can see from my small tiles what was the stitch result. So this is what I get at the end for the fluorescent channel. I can also check what was the result for the phase contrast. And this is what I have here. And I can also check what was the result for segmentation. But what I want to do also is to overlay all of that. So same thing as before. From here, I can specify different layers. So I specify that I want my fast contrast layer here, my fluorescent layer here, and then I want to overlay also the segmentation. And I put a hot color map to the segmentation so we can really see the different segments. Okay, let me put that full screen. So this one is my fast contrast image. So next I want to overlay the fluorescent one. So I can see it here. Again, I can adjust the transparency between the two channels. And then finally, I want to overlay my segmentation. So for example, I can zoom in here and I can kind of play with that to see is my segmentation accurate or not. And based on that, I could rerun and adjust parameters. Okay. And then in terms of more AI-related workflows, so I'm going to show you two simple AI-based workflows. So the first one will be training and inference with our unit-based plugins. And the second one will be segmenting nuclei images with StarDist. So let me show you the data first. So we do have some RP cells. So RP stands for retinal epithelium. Let me not butcher that. So we have for our group, we have a website that is called asg.nist.gov. And what we do on this website is we publish data. So anything that we do in the group that is then published, and we publish the data set here. So you have a lot of data that you could download if you wanted to work on similar topics. And so what I'm going to show here is extracted from these two set of data sets. So we have the retinal pigment epithelium cells, and then we have 2D and 3D images. So to start with, I'm going to talk about data set that I downloaded from here. So I downloaded the fluorescent images and then the manually segmented images, so my ground truth to train the AI. So let me show you this data in the system. So then I downloaded all of the data and I separated them into training and test data. So I kept like 80% of the data to do to the training. So if I have the fluorescent channel here, I can adjust the contrast so we can see something. So this is basically what it looks like. Small images here. And then I have the matching mask. Let me show you the masks that were created by someone's domain expert. If I wanted, I could invert, so it might be easier to see on the eye. And then I have some test images, so different from the training ones. that I want to use in the inference part of my workflow. So let me show you what that would be. Yeah. So this would be the workflow that I would create. So the first one, we use the UNet CNN training plugin. It is available on our GitHub as well. So when I say our GitHub, it's US NIST Gov organization, and then you can search for all of this. Everything that starts with WIPP, most of the repos are WIPP plugins. So I specified what were the images, what were the masks. I specified no tiling because they were already tiled. The training fraction, batch size, I only put one here to not overwhelm some of our servers, number of class, and all of the information that we can add to this plugin. I specified, for example, that I wanted to use augmentation, so I could get more training data as part of the training process. And as an output, I got a TensorFlow model. So this one is not running yet, but I would get a TensorFlow model, and I also get TensorBoard logs. And then my second step would be to want to use the cells that have not been used for training, and then the model from the previous step. And then I get this two-step workflow. So I could submit it and monitor the execution. But because it has a training job as the first one, it might not be done by the end of this presentation. But we can look at, for example, some of the logs that would happen. You see a bunch of TensorFlow messages. So it is starting to load everything and to prepare for training. So if I show you, I have it decomposed at two separate workflows here, for example. So this is the same thing, just training. We still see the same parameters, and as an output, we see the TensorFlow model that is here. So basically, we can just download it, and we can also look at the logs from TensorBoard. So yeah, so this is kind of integrated with our platform as well. And then from there, we used it as part of an inference plugin. So in this inference plugin here, I took as an input my test cells and the train model from the previous step. And as an output, I will get a collection, and then we can check the result that is here. So this will be the result of my two-step workflow, training and then inference. And then we can look at one last type of workflows for nuclei segmentation. So let me show you the original data. I think it's called, yeah, it might be this one. So this one is also coming from our kind of repository of data that we have here. So this is from this one. So this is from the 3D dataset, but we split the stack into a set of 2D slices for this demonstration. So, but as you can see, there are a bunch of zip files that can be downloaded with all of the original data. So we can see here, so yeah, some nuclei images. And what I want to do to segment them is to apply a package that is called StarDist. It's kind of well known for nuclei segmentation. So what we did is we created a WIPP plugin for that based on the Python library. Yeah, and what we would do, I think it's this one. We would just select that we want to use the StarDist inference plugin. We will specify our input images. We select which pre-trained model. So they offer a few models. So here we have fluorescent image, so we want to use a fluo model. As an output, we will get the mask from StarDist. And if I do some contrast enhancement, then I can see my masks. And again, what I would want to do is to overlay both images so that I can check the result. So I can do that here. and I have it prepared here. So I specified a first label with the fluorescent images. So this is what we were looking at initially. And I can specify my pattern. So TTT stands for time. So this is kind of what is varying in the image file name. So this is what allows me to have multiple time frame here in the visualization. So I can specify that. For the fluorescent, I did some contrast touching, and then I applied the color map hot. And for segmentation, I just did the contrast stretching. So if I collapse that and make that a bit bigger. So we can see here the raw images with some color mapping here. So we can get 2 different colors. And then I can overlay the segmentation, and I can check if I'm happy with the segmentation, which look pretty good here. But yeah, this is useful to check segmentation results. Okay. We could check, yeah, but the training one is still running. Yeah, it takes about 10 to 15 minutes, so we might not be able to see the result. Let me go back to the presentation here and check if there are any questions in the chat. I don't see any question. Okay. So let me move on a bit here. Yeah, so this is, we also have, so on our GitHub, us.nist.gov/WIPP, we have some kind of a few tutorials, I would say, with some test workflow. So a workflow really similar to the first one I showed is described as part of this README files. And yeah, so this is just some explanation about the images that I showed for the unit segmentation workflow. So it was using images from this project, from the retinal pigment epithelial cell project, and kind of to kind of have a simple explanation on that. One of the goals of this project was to be able to predict whether an implant, so this is for implants in the eye to cure macular degeneration. And so to predict if the implant is healthy or not via imaging, as opposed to more kind of destructive measurements such as transepithelial resistance that is used. So in this kind of project, you can learn more on the isg.nist.gov webpage, and you can also download all of the data from this project. And so this is what we did. We did a 2-step workflow with training and then inference. And then so from these images, we then got the mask. And then the last workflow that we did just before was the StarDist one. So we got, again, some images from the retinal pigment epithelium cells project. But this time we were looking at the nuclei. So we used StarDist, and we got this type of segmentation. And we were able to overlay the raw images and the segmentation and kind of compare the 2 sets of images. Okay, let me check if there are any questions regarding the demonstration. Okay, so moving on to kind of the last part of the talk, just to show you what we are currently working on that is not in the platform yet, but we are hoping to release in the next few months. And this is about reducing human annotation efforts through traceable iterative AI-based retraining. And so one of the first steps for that was to add AI model cards in the WIPP platform. So right now, I don't know if you remember, but when we got a train model in WIPP, so it was registered in the WIPP platform, but it had really minimal information, just a pointer to the training job, a pointer to the tensor board logs, creation date, author, things like that, but not much more. So we decided to add model cards. And we try to make the model card model that we use as generic as possible, so we could then export it to different formats. So for example, if you were to train a model in WIPP and then you wanted to export it to Hugging Face, Bioimage.io, or some of the TensorFlow repositories. So we are building some helper for that. And also CDCS, which is our project that we use for the WIPP plugin registry. So we want to also be able to store train model in this website so that we can quickly search for pre-trained models that might fit our needs. So we don't need to retrain all of the time from scratch. Yeah, so and we are enabling AI model upload. So if you already train a model outside of WIPP, you may be able to upload it into WIPP to reuse it. And we're also adding the TensorBoard logs display directly into the web page as opposed to having to go to the actual TensorBoard dashboard. And in terms of how we enable reducing the human effort, so we are building interfaces with other platforms, so notably image annotation tool platform. So in this case, we started with CVAT. So it stands for Computer Vision Annotation Tool. So this is a well-known platform for image annotation, and they have an open source community edition in addition to their more paid versions. And you can do 2D and 3D manual annotation inside, but they also have automatic and semi-automatic annotation capabilities where you can use pre-trained AI models directly in the platform to bootstrap your annotation. So this is really useful. And so what we are doing is to integrate our WIPP platform with their CVAT platform so that you can really go back and forth between the two, share the data storage, so you are not copying data over all of the time. And then we can re-import annotation into WIPP as masks, for example, to use to retrain AI models. And the way we are doing that is by building an annotation API that we call, that is really kind of middleware between our WIPP platform and the CVAT platform. So the idea is really, you have images in WIPP, they are being sent over to CVAT, but in reality, they are just being loaded from a shared drive. So we don't duplicate the data. You can use all of the cool features for annotation that are in CVAT, and then when you're done, it will automatically, through webhooks, trigger the import back into WIPP, and then you can retrain and go in a loop like that until you are happy with your train model. And so one of the examples that we use as part of this project is this one, so some concrete images that we got as part of an old project. So this is images of concrete from nuclear plants. And we had someone that was annotating, like a domain expert annotating different type of defect that we can see in the concrete. And so what it would like with integration, so you have your concrete images in WIPP, you select different types of labels that you want to have as part of the annotation process. You can assign different users and say how many images per user you want to have. And then it will be sent over to CVAT, and this is in CVAT what you will see, where you will be able to then use some of the tools. So let me show you quickly. So our custom instance of CVAT. So this is kind of the same type of images that are now in CVAT. And one of the cool things that we can do with CVAT is you can integrate pre-trained models. So one of the models that they offer is SAM, so Facebook Segment Anything model. So for example, here I could start, let's say if I want to segment air void, and I'm going to assume this is an air void. I want to use segment anything, so I click here, and then I can start clicking, and I will get, so it's a bit, yeah, it's a bit slow the first time, so I can quickly segment this part. I can say I'm done with this one. Next, I want to segment, and here I'm not a concrete expert, so let's say aggregate damage and I'm going to say that this is aggregate damage, but I'm really not an expert, but just for the demonstration, you see it's quite easy to segment things with some. And then I could say so I'm left click on what I want to keep, and I could do a right click to say, no, this one is really not part of it. So that's what I just did. Yeah, so this allows to really go kind of faster to generate segment annotations, and then the user can always go back and adjust what was automatically segmented by Sam. Exit that. Yeah, and I think that's about it for this part. Okay, and as I mentioned, so if you have any questions, or if you want to get the presentation or something, you can always shoot us an email at wippteam@nist.gov. So this will go to me and other members of the team, so we can answer your questions. And then we have here. Yeah, so we have us.nist.gov/WIPP on GitHub. This is where we have most of the documentation about our WIPP platform. Yeah, we have some list of plugins here. And some user guides, as well as tutorials. So tutorial should be here, yeah. Okay, let me just check if there are any questions. I don't see any question for now. Yeah, so if we don't have any questions, thank you so much for joining me today and listening to this talk. And please don't hesitate to send us any questions at this address. I see something in the chat. Can you show us how to install the WIPP? So basically, this is kind of meant more to be installed on a server or on a cluster or in the cloud. So the installation is I don't want to say involved, but it's not really something that is going to be installed really fast on a laptop or desktop, for example, because it relies on Kubernetes to run the computational workflow. So we are thinking about maybe releasing a lightweight version that will do just the visualization, for example. So you would not need to install as many kind of Kubernetes tool and Docker tools and all of that. But so we do have some installation instructions, including kind of testing instructions. Let me check. Yeah, this one. So where is the installation? Deployment, yeah. So we have some test instruction where you would be able to use maybe Docker desktop or multipass with micro Kubernetes if you wanted to test the system. But yeah, it's mainly meant to be installed on the server that has Kubernetes already installed. So it will be basically running a bunch of Kubernetes command to install it. And we do also for those of you who are familiar with Kubernetes, we are developing some Helm charts for WIPP, so it will be easier to install on Kubernetes cluster if you're already using Helm, because then it's just a few commands to install it and configure it. Hopefully that answers the question. I see another question. Does it support other file formats beside omitif? So basically, what is supported by bioformats would be what we are able to ingest. But so in terms of ingestion, whatever is supported by bioformats will be supported. We might have some specific formats that we have to tweak or something, but usually what is supported by the bioformats, we are able to ingest. Then in the system, it will be stored as omitif. And when you do computational workflows in WIPP, each step you get, if you get an image collection as an output, it will also be converted to omitif to get the scalable visualization. What we have been looking at is also omizar, so the ZAR format. So that's something that we are planning to enable also as an option for the conversion. And also we are thinking about having a raw option where you would say, please no conversion, just ingest my images as they are. But then you might lose some of the visualization tools. So yeah, so that's the answer. Have you guys tested three-dimensional data? So yeah, so we do have some 3D data set. Right now, mostly we use them in the omitif format. But as I mentioned, we were also looking at ZAR. And I know we have some collaborators at NIH that are using the ZAR format a lot. As the uploaded images saved on the server that WIPP is installed on. So it depends really on the installation. So if it's on a traditional server and then you have data kind of sitting on the server, then yeah, it will be saved on the server. If you're in the cloud, then you can use persistent volume and it could be stored depending on how it's configured on another node or another type of storage. Yeah, so we do have to have the data available. So if you have a multi-node installation, we do have to have the data available on all of the nodes. So it really depends on the configuration. But with Kubernetes, it's kind of easier to configure that. Yeah, sure. Thank you for your questions. Okay, we have about 3 minutes left. So any other question I can quickly answer? I don't see anything else. So okay, I'm gonna leave 1 more minute just in case. But otherwise, yeah, thank you so much for listening to this talk and don't hesitate to contact us here if you have any questions or interests or comments. Thank you. Okay, I think there are no more questions. Thank you so much. Bye-bye.